Okay, good morning, everyone. A warm welcome to all of you. Let me start by naming some milestone years. 2009, 2014, 2015, and 2022. Why naming these years? These are years that bonding us as parents of a child with a special need. 2009, it was the year my son Cedric was born. And in November, I had my very first consult at genetics. 2014, the very first publication came out, an ADNP on autism-related syndrome. And in 2015, the syndrome was named. 2022, it was the first time we had our European Congress. So therefore, I would like to ask you to warmly welcome one of the, if not the key person in the discovery of the Hels-Moortel van der Raas syndrome, also known as the ADNP syndrome. So who other than Dr. Nathalie van der Raas is best placed to give you more insight in the history of this syndrome. So please, a warm applause for her.
Thank you, Kelly, and thank you everyone for being here today and granting me the honor to speak to you about how we found this syndrome. So, I just want to explain to, if I may, how syndrome identification, how it works. And I'm going to explain to you what a syndrome is, and then how we used to define a syndrome in the past, and then what novel technologies brought when I was in the genetics department in Antwerp, and how we were able to discover this new syndrome. So by definition, a syndrome is a set of developmental abnormalities that occur together in one person, and the pattern that it occurs is recognizable. And we assume that it is the result of one single etiology. So there's one single cause to all the abnormalities or the developmental anomalies that we see in our patient. And if there is a cause that leads to an error in what we call the morphogenesis, so how the development of what we look like and what the body, how the body is formed, is there is a cause to that, we speak of a malformation. And if we speak about dysmorphic features in the face or in the body, it means that there is an abnormality of structure. And this can be a visible abnormality of structure in the face, but also in an organ, like in the heart or in the ears. And in order to know when something is abnormal, we need to know what normal embryology is. So I came to clinical genetics as a pediatrician in the year of 2005, and it was a new era to a lot of novel technologies. Before that, in the years before, we would first describe the syndrome and then look at what the genotype is. So from phenotype to genotype is how we used to work. A phenotype means what we look like. So how our face is, how our body is our phenotype. And the genotype is what our genetic information says. So we used to work as as pediatricians and as clinicians from early on in the 1800s by looking at patients. And then when we saw a recognizable pattern, we defined the syndrome clinically. So one of the first well-known syndromes, or the first well-known syndrome that was described was in 1862 by John Langdon Down, known as Down syndrome. And then nearly 100 years later, finally the cause of this Down syndrome was identified when people could go and look at chromosomes in the cells containing the genetic information. The cause of Down syndrome, as you can see here. Okay. Yeah. So, sorry. Chromosomes. Every cell has 46 chromosomes, and every chromosome comes in twofold. There is one copy that is inherited from the mother, the other one is inherited from the father. In Down syndrome, it was found that these children had 3 copies of the chromosome 21, so trisomy 21. So we looked at the phenotype, what does a child look like? And then this is a physical expression of the genotype, and then we processed the techniques to look at the DNA. So from the 1950s, 1960s, that was mainly looking at chromosomes. From the 1990s, we were able to look very smaller than the chromosome, to look at parts of the chromosome, either by fluorescent techniques, coloring techniques, or other. So, and that's what I've always worked. That's the way we always worked in my training as a pediatrician and as clinicians. When I first started, we looked at a child, and then we went to look for the confirmation in the genetic material. So another very well recognizable syndrome was a deletion where a part of a chromosome is missing, and we found this by in the early 1990s that this was due to a missing part that we couldn't see under the chromosome, but then the fluorescent techniques, novel techniques, brought the possibility to diagnose this. And then later on, we were able to look at separate genes and to examine by special techniques one gene at the time, to go and look for mutations in that one gene, or to see whether one gene was missing. And I have from the early 1990s, from 1993 up until 2007, with this way of working, a lot of diseases were able to be tested. So testing became available over the following 15 years for a lot of genetic diseases or a lot of syndromes that we saw in our clinics. Now, a lot of syndrome patients remained undiagnosed, because we saw some quite unique cases in clinic that we couldn't recognize as a known syndrome, or we saw some children that resembled a known syndrome, and then we did the tests and we couldn't confirm it by the genetic testing. So the genetic diagnosis was very challenging, also because even if a syndrome is known, there can be some variability in what the genetic cause is, but also there can be some variability in how the patients present. So that makes this type of work quite difficult. And by the time I was in the genetic department, we were sort of left with a whole pile of unknown cases, and we kept on following up these unknown cases. Sorry to call these cases, patients where we didn't know the diagnosis, of course. And there was quite a few, and we followed them up on a yearly basis. meantime talking to the lab about what novel technologies came up and trying to find novel ways to help confirm a genetic diagnosis in these syndrome patients. So between the year 2000 and 2010, we could use in the group of patients two novel technologies, the microarray, which is a way to look at the chromosomes in a very small parts. In the Department of Clinical Genetics for Developmental Problems, we've been using it since 2008. And then in 2010, we were able to use on these patients where we didn't know the genetic cause of their syndrome, the whole exome sequencing. So we changed a little bit in our way of work because historically, first we looked what is the syndrome clinically, and then we go and find the genetic defect in the patient. Whereas now we have the DNA, we go and find the genetic defect, and now we go and look, what is this syndrome then? So it was working the other way around. And it's called the genotype first approach, where we look at what the DNA says, and then we go and see what it means. So we saw, using this genotype first technique, we saw an increase in the percentage of diagnosis, of course, and we were also able to identify novel syndromes. We were also able to identify new genes for disease, and we were also able to identify risk factors for intellectual disability as well as autism. So this story of Kelly and Cedric begins in 2009, where I see this cute little boy with representation of global developmental delay and failure to thrive. He was not growing well. And examining him, he had a heart murmur, and we found a cardiac defect, a hole in the heart. So he was sent to the cardiologist, and that was treated to improve his growth, because I knew sometimes holes in the heart can cause failure to grow. But still, there was more going on by the look of his face, and also the look of his MRI was not completely normal at the time. He had some lesions on the brain scan. So I wanted to look further into this. I looked at in the undiagnosed cases, so we usually looked at the chromosomes in general. We did an array to look at the chromosomes, very small pieces of chromosomes, and we couldn't find any abnormalities. And then we have to start thinking, is this one specific syndrome, or what could this be? What could this look like? Can we do one of those specific gene tests? And with the cardiac defect and this facial feature of Cedric, I wasn't really sure, but I thought maybe it's an atypical Noonan syndrome. So let's target on the genes that can cause this syndrome. And these revealed no mutations, so there was no diagnosis. So if we have no diagnosis with the used techniques in diagnostics, we walked to the research lab. So I walked to Frank, and I spoke to Frank Kooy and his researchers. And I said, what can we do to find a diagnosis in this boy? And we had Celine Helsmorstel as a student, as a PhD student, who was going to look at some next generation sequencing, who was going to look at some full DNA sequencing results in 10 of my patients. So Cedric was a good candidate for this approach. And what she did was she took DNA from the father, DNA from the mother, and compared it to DNA from Cedric, from the patient. Just have to make sure. So what we do then is DNA in the lab just looks like a following up of letters, which are base pairs. And we know what the normal code of DNA is. We know when there is changes. So sometimes we see changes that don't mean anything. Sometimes we see changes that do mean anything. But when we compare the child with the healthy parents, we can find out, is there any changes in this child that are not present in the parent? Because as the parents are healthy, we assume that the changes in the DNA we see in the parents are not disease-causing, whereas the change that is not present in the parent is disease-causing, maybe disease-causing. So, and that's what's pointed out here in the DNA of the child. It's called a de novo mutation, so it's not in one of the parents. And without going into the specific technical specifics, usually we find in a lot of these patients 1 or 2 de novo variants. This doesn't always mean that they are disease-causing. So in the DNA of Cedric, we found this variant in one gene, the ADNP. It was a 4 base pair deletion, which means that there's a tiny little part of 4 base pairs missing in the DNA. And what we do then is we look at the DNA of loads of populations, of thousands of people, which is a control database, and we go and check, has this been described in any healthy person? So this mutation was never described in any healthy patient. So then we go and look, what does this gene mean? What does it do? What does it do? Can it be something or not? So, and we found out a little bit about it, also through the work of Ilana Gozes, who was working on ADNP at the time already. And we know that it's very important in the brain. It's expressed in the brain, and it's involved in what we call neurogenesis, the whole development of the nervous system and the brain. Ilana found that when we make mice that don't have this gene, they die. If they don't have this gene at all, if they have only one working gene, that they have problems. So I thought we might be onto something. This might be a possible diagnosis for the problems in Cedric. So then, of course, only God knows. I'm not God, but what can I do to find out? We go to network meetings, we talk to colleagues, and we ask everyone that's in developmental genetics, has any one of you seen any child with genetic mutations in ADNP? So everyone has gone to look at their material, people from Sweden, from the United States, from France that we were working with, from Holland, from the Netherlands. And we were joining conferences on novel intellectual disability syndromes to present this patient, Cedric, and to ask for more information to clinicians and other geneticists. So, and people started sharing plans. Yes, we have one. And we ended up getting 5 patients at the start, and then later on, even 10. And these are the first 8, sorry. These are the first 8 patients that we found. And we found some similarities in the face, some similarities in what we call the phenotype, the way they present. So these were also listed up. So as you can see, There's patients from all over the world, also from Italy, from Seattle, from Stockholm. Maybe one of your children is in our first paper. And we could list some syndrome characteristics by looking at the clinicals. And that's the way that's why we decided to publish this novel syndrome, to have clinicians be on the lookout for it, because this gene might be important in intellectual disability, autism, and some characteristic syndromic features. So, Celine did all the technical work, Celine Helsmortel, who's not here today. And she was the first publisher, and I, as the clinician, was the last publisher. And then, as Kelly mentioned, the year after, the syndrome got a name, and that is not Celine and me that decided that this should be the name. It's the way OMI works. OMI is the database of genetic diseases, and there is some algorithm for finding novel syndrome names, and apparently they picked the first and the last author of the first paper, so that's why the syndrome got her name. And after that, the network of developmental disorders, which is led by Frank Kooy and Bert de Vries van Nijmegen at the time, they wrote a web page on the ADNP gene, which led to the, may I say, the explosion of the case reports. So we're very grateful also to the team of Nijmegen and the European Network for Developmental Disease Recognition. And that's my talk. After 2014, Anke van Dijck continued the clinical work. She'll be here today later also to speak to you. And I'd be happy to take some questions if there should be anything unclear to you.
Thank you, Nathalie, for your talk, for your explanation of the syndrome. Any questions for Nathalie? Okay.
I was wondering how many patients are now recorded with the ADNP syndrome?
The last records, Frank, are, what number do we have now for this?
There's no registry.
There is no registry.
I think Kelly knows better than.
Kelly knows better. 78 is on the ADNP website in here, but there is more.
I can mention 150, something around that. Okay,
The American website, there may be close to 500, but I don't, maybe it's very optimistic.
Yeah, it's
So it's very optimistic.
it's not,
But
yeah,
I think the calculating,
not all confirmed.
not all confirmed, okay, so
Remains unclear apparently.
From the last things I know, we have 94 diagnosed cases.
94 diagnosed cases. And 78 registered in the ADNP gene registry. Okay, so put 16 in them.
Okay, any more questions? No? Okay, yeah, Claire in the back.
Is it already included in the gene panel?
Yes, it's included in most of the developmental disease gene panels, as it's now the 5th cause of syndromic autism and developmental disability. Yeah. Yeah.
Okay, no more questions then? Okay, thank you very much. Then we move on to the next speaker, but let me just have a seat, because I think it is Professor Frank Kooy. And he will give an explanation about, and then I need An update on the clinical presentation of this syndrome. Okay, great, go ahead.
Just, on behalf of the very warm welcome here. I will let you continue. I should actually doing a little bit of Anke's presentation. Actually, I'm going to continue where Nathalie stopped the story. So I'm going to explain a little bit about the clinic, if I can get there. Without that's me. By the way, we are recording this, no promises. We're doing our very best. So and hopefully, and the idea is to make these recordings available to you all on the website. There we go. Yeah. Why this first slide is actually a continuation where Nathalie stopped. This was we had a consortium to work on ADNP because when you find the gene, you're very excited. You say, no, we have the cause, but then of course, then you all come and say, okay, so what can we do for you? And therefore therefore you need funding. And one of the fundings we got was actually fairly prestigious, was from the European Union. So we got a so-called European consortium. You'll recognize Anke was already there. There's more people. Oh, Sandro is in the audience here. He was a PhD student at that time. And it was actually a recording just made very close by here. Anyway, the Harutyunyan from the RAS syndrome, something about the name, by the way. It's basically no different from why is it ADNP syndrome, why is it Harutyunyan from the RAS syndrome. It's a bit like you can call yourself any name you want. You go to your life, you call yourself, your first name is Patricia, you call yourself Lily or whatever, no one cares unless you go and you walk in and you want a new ID, then you have to give your official name. Same here, you can call the syndrome, of course, these are your children, anything you want. If we publish, we're bound to use the names of the OMIM, the central website, that's a registry, they call it Harutyunyan syndrome. So we should call it like that. Sometimes it slips through, I've noticed with ADNP syndrome. I don't know why or what, but that's the story behind it. This is a very complicated slide, but basically, this shows you if you have a large population, if you're speaking of 10,000s of people with what Nathalie described, a form of syndrome, mental handicap, and we don't know what it is, then we see. And we look all over the genome, what could be the cause. Then we find mutations in all of these genes. Because in this publication, there's 72, there are more, but in this publication. And you see that here, we gave it an error because you can't read it, of course. Is it ADNP here? It's one of the most frequent ones. Depending on the study, it's always frequent. That's not so much why I show this, but why I showed it, and that's also part of the answer why we're not whether some databases give you many more patients than other ones, is that if you look here, if you see the color red, it means that if you do genome-wide screening, you only find mutations that take away and that destroy the protein completely. That's weird, isn't that the case with all syndromes? No, usually you also have the other colors. Sort of errors in the DNA that make a change in the protein. So you have the protein that is altered. There's no evidence, at least from the mass screening, we never see them for ADNP. Doesn't mean they don't exist. We're working on them, but they may be there. I think some of the databases are very liberal. They take those other sorts of mutations. We are usually a little bit more strict, and we only say this is Harutyunyan syndrome if we're 100% sure. Doesn't mean the other ones don't have it. We just don't know. What?
Zou je meer in de microfoon spreken?
Sorry?
Zou je meer in de microfoon willen spreken? Wij horen u niet.
Yeah, but then it starts echoing. Oh, you. I'm not gonna. Is this better? Yeah. I don't like this type of fixed microphones. But anyway. What Anke actually did was, and I'm sure many of your children are there. Wonderful. What Anke actually did was take a large cohort of patients, and many of your children may have been in, and then gave all the list mostly to the clinicians and said, what exactly, what features do you have? And of course, you want to do a comparison, so you don't ask, what do you have? You just give you a list of like, I think 150 issues, length, height. You don't, it was a long list. It was actually, and then collected a group of 78 children. At that time, it was the largest cohort, one of the largest cohorts for rare disease ever described. You see here's where they come from. Most of them European, a little bit USA, some of the other countries. You see, from within Europe, everywhere. You see, most of the children were young. This has to do with the way they're diagnosed. That's what we can speak about. We have a reasonable insight now what this syndrome gives when your children are young. When they're older, we don't know at the moment because we haven't seen them. Doesn't mean they don't exist, but it's just a way, because you need both parents for a diagnosis at that time. That's a selection for younger against older children. Of course, here you see, and you may recognize your own children over here. See, this was a list of all the children. And here we can see that some of the facial features that we noticed, like Nathalie and this one, that are actually common. Of course, not all of your children have all of the features, but as a group, yes, we see these features. And then, because primarily the emphasis was of course on the intellectual disability and the autism, but then we also asked for cardiac abnormalities, for behavioral problems, for brain MRIs, for feeding, for visual problems, and there was you see the and these are the percentages in autistic features. You see them in that's up to 93%, depending how strict you have your criteria. Cardiac abnormalities, behavioral problems, brain MRI. But you see, these are all maybe associated with the syndrome. Now, one thing that I want to point out, that if you see cardiac abnormalities, and Cedric is an example there, they can be used of all kinds. So it's not like this specific abnormality. There could be basically anything. It's hard to understand some of the feeding problems, visual problems, brain MRI. Basically, it's not like you have light ventricles all over the place, like in some syndromes. It could be large ventricles, could be corpus callosum, could be anything. Could basically be all of it. Now, the other parameters, you don't have to look very carefully. I'm just going to go through, but like birth weight, birth length, etc. That's all. See the dots here, male, and here these are individual patients. Nothing seems to stand out. It seems all normal. Weight, they're a little bit over average. Length, a little bit under average. This is all for, again, you see a setup against the age again. We don't have any data don't have any older patients, so we don't know. So otherwise, pretty regular. So this is the clinical, this is a clinical summary. So a lot of general health problems. See them all here. Feeding and intestinal problems, visual problems, etc. So they're all in decreasing order, brain abnormalities, musculoskeletal abnormalities, cardiac defects, etc. So apparently it's much more, as you are pretty much aware. I'm not much more than than just an ideal. What's new here? This is something that I would like to bring up. Which I would like to point out. It's under development, as we say we say, so it's under debate. But of course, we all have different mutations. You see them there here. If this is a representation of the gene, if you see all of the mutations, see them across the gene. And some of them are more frequent than others. This is for instance, the PTER mutation is actually quite relatively frequent. It's the most prevalent mutation. So we were of course interested in this was to see if we compare the different mutations with all of your clinical presentation. Does it have an influence? Are you different with one mutation from the other? We don't know as yet. We did. Anke initially did it, and we found only with this mutation, we found that if you look at like walking independently, patients with this mutation were a little bit later on average. There were a few more, but otherwise there were very few differences. So there was a hint. We were a little bit hesitant there to bring it out because you do the statistics, you find it. You can't say we didn't do it. Well, quite convinced, but then things change. Research developed, and if you look here, you see two protocols for the mutation. It has to do with another technique that was developed, so-called methylation analysis. I'm not going into the details, but what it says basically says you can discover, if you look at other characteristics, two groups of mutations. We weren't aware, and when Anke wrote out this paper, we weren't aware of these two classes of mutations. And also time develops, and as the next phase, all this morphology was done by clinicians looking at and discussing with each other. Time evolves, and our colleagues in Nijmegen, Tingemans, PhD student Verbruggen, they went on, they took all the photographs, and they did an AI-based algorithm, and they could sort of make. And now the computer, believe it or not, you show a number of faces, the computer can recognize, hey, this is likely this syndrome. Very good, very clever. And they also, but they don't only judge the eye, they only look at the other, at the interview with the patient, with you as a patient, and they look at the other abnormalities that are described. You have the ID, you have the hypertonia, etc., etc. And then combined, they can say, hey, this is likely, based on the algorithm and the interviews, they can say you've likely this and this syndrome. It's quite revolutionary because if you go to a clinical genetic center, you're likely to get the right diagnosis. If you go into more distant
skeleton. So we have a skeleton as humans. It keeps us like lifted with a nice posture during the day. But the cells, they also have a skeleton, and it's really important that cells have the correct shape to communicate with each other. And we see that the skeleton of the cell in the mouse is not as strong as it should be. And the combination of these two things make it a little bit more difficult that the mice have cognitive abilities. So the mice have problems with learning. Now, I want to point out this slide because over the last couple of years, we have been working a lot with clinicians, scientists, patients, and parents. And this connection is very important. And we should always put emphasis on this strong connection because communication between you and us is very important because you are the first line guardians and you can teach us more probably than we can teach you. And in this way, it's also nice that, how can I say, if I go to work, I don't always have a great day. Sometimes I also have a little bit of a day off. But you guys give my work a little bit of meaning and purpose. And when I have a bad day, I always think about you guys, and then it's nice to go to work again. And then it's nice to work with these sometimes very not so pleasant mice. For the last couple of years, I've also been an advocate for this syndrome, and I was also involved in creating awareness for this syndrome. And I have entered lots of competitions for science speaking, and I've also been, I was lucky that my work got featured in EOS Science magazine, in which we create awareness on how this was discovered. And Kelly, this was your story. And I was very fortunate that we could together be published in this story on how the syndrome was discovered, how we do the mouse work, and how testing of and keeping on testing of these genes can maybe be can give a little bit of hope to all parents. And then also, and that's something that still today strikes me, it's the initiative of the German Patient Foundation. There was a crowdfunding, and within 1 week we were raising over 30,000 euros for scientific research. And I can honestly say I have never seen this for any syndrome in my life, that in 1 week that we could raise so much money. So this is really, really nice. And I really want to thank you again for it. But of course, you guys gave me a lot, so I'm also here to give you something back. And last time I talked a lot about mice, but today I want to talk about children. And we call this in the clinic patients. It's how can I say, we call it patients when we communicate with our colleagues. Please mind that we are talking about children. For example, our first case study, you see here a very nice, sweet little girl. She was 5 years old at the time. She had intellectual delays, developmental delays, and now she has also been diagnosed with autism. There were speech and motor delays, and there were some slight alternations in the face, but they were not as prominent as with the vast majority of children that we see in the clinic. What was also very striking was that the teeth of the children were lost before the onset of 1 year. And then Frank gave you a nice introduction how we incorporate AI software within our lab and throughout collaboration. And we send pictures, and that's why we always ask pictures from the children. We send it to the AI algorithm, and then we get a score. And you get a score based on the facial appearance, and you get a score on the, how can I say, the symptoms that are presenting. With the face, we get a score of 0.85. It's close to 1, which means that based on the photographs, this child was very strongly correlated with the population of children with Helms-Mortel van der Waals syndrome. So it was a really good fit. However, this was counterintuitive. When we look at the clinical features, it was not as strong, not as penetrant as we would like to be. But back in the time, there was also, like I said, autism was, for example, later diagnosed. If we incorporate it now in the algorithm, it would be more dominant. But nevertheless, we had some evidence, but we're on the right track, but we want to be more sure about this. So the parents consulted with the clinical geneticist. They did whole exome sequencing. And in the whole exome sequencing, no diagnosis. Okay, we'll go to the next step. We do a whole genome sequencing, no diagnosis. And then, very strikingly, and this is a very new technique, when you look at the methylation of the DNA, so it means like you have remembered the genetic code, you can put flags on this, and when you have a special mark of flags, this is a signature, and you can look for this. And from this episign, there was a de novo ADNP mutation. We were like, oh gosh, how could we miss this? So we went back to the whole genome sequencing data. We looked into it, and actually we were a little bit too stringent for the data. We said like, no, this is too harsh. But when we lowered the threshold, this mutation was also present in the whole genome sequencing data. And the mutation of this girl is a very special mutation. It's a unique one. It's one that has never been seen before. And if you look at the ADNP gene here, you see all these blocks. We have 1, 2, 3, 4, 5, 6 blocks. But only the last 3 blocks, they form the mature ADNP protein. And you see this arrow here. This arrow is where the mutation is happening. It's right before the start of the ADNP gene. We did also the Sanger sequencing to confirm that the deletion is really present, and we see that in fact it is present. But the site where this mutation is occurring is very special, and please let me guide you through this. So I said that ADNP envision it as 3 blocks, and these blocks are connected with lines. And it's like shooting a movie. If you're filming, you keep on filming, and then you need specific scenes, and you have to cut them out and paste to each other. And that's also how the cell works. So this is a movie, and then normally we don't like this bar, we cut it out, and we paste everything together. That's how it's normally done. What can happen if the mutation is inside this connecting line? The cell sometimes does not like this, and sometimes it skips. So you see that only the number 2, it's not incorporated, it skips it. Not interesting. Sometimes the cell can recognize it and it can include it. And sometimes there are also combinations possible, but this is a little bit too difficult. But now we have to find out where this mutation is happening, and it's happening right before the start. So it's happening just here, and it could be that one of these mechanisms could be in place. So we took bloods from the parents and the child. We did transcriptome sequencing, as we call it. And then you see here that the mother has a bar, the father has a bar, but in the child, this bar is absent. And what happens when we look in our computational data, the cell does not recognize the start anymore, and it goes directly from this block to this block. It skips it. So it skips the start of the first block. So that means that the start is essential for the formation of the protein, so it's not formed. But we have to prove this, of course, and therefore we clone the gene in an expression vector in bacteria, and the bacteria learned us that there is no formation of a mutant mRNA, and there is also no mutant protein, so the protein is absent. So as a result, the child presents with one normal copy, and the other copy is basically ignored. And we call this, in difficult words, haploinsufficiency. And we were very fortunate that we could publish this unique case in the European Journal of Human Genetics. And this was, I think, a very nice way in how the initiative actually came from the parents, and me as a scientist, as a team, we went looking for the cause. So I really want to thank the parents, especially for making this precious donation. And together we have put out this nice work. And that's the first story that I would like to tell you. For the second story, I want to say before I want to speak about it, it's a difficult subject. And we were doubting a little bit to speak about this, but we've consulted with the Patient Foundation, and we think it's quite important to mention this. And I want to say that this study especially, it's very close to my heart because it is an invaluable contribution of a young child that has been passed away with Helms-Motor syndrome. And the findings of this study stem directly from this remarkable donation. It tackles a very challenging subject, and it's organ donation, because the parents of this child that has passed away, they have donated almost all the organs to us and to ensure that we could investigate it. And it's very important that we have done this work because we do a lot of work in mice, but now we had really child material in which we could see if what we do in mice is really also the case in humans. And I would like to express my deepest gratitude for this family and their courageous donation, of course. This is Vit, and Vit was 7 years old at the time. He presented with autism, developmental delays, and he had severe epilepsy. He has unusual facial characteristics, as you can see on the picture, and also his MRI scans were not that great. He received a combination therapy of anti-epileptic drugs during his lifespan, and he also underwent 2 liver transplantations. Following the second liver transplantation, he unfortunately passed away. His parents donated parts of his body to us, and then we went into the lab and did a very clinical and molecular investigation. And as I mentioned before, one of the easiest things that can be done in the lab is study the methylation, because methylation is quite robust. We can even investigate Neanderthals with methylation effects. It's very robust, and we did it in the brain. And we saw that the mutation in ADNP of this child causes abnormalities in methylation. And this is very, very nice because we see that it is also happening in bloods, and we see it's also happening in the mice. So it's a very nice correlation that the findings that we have in mice and in the human bloods, that they're also present in the human brain. It's a very unique feature. And when we look for the processes that are dysregulated in the brain, we see that the structure of the cell, the cytoskeleton, is affected as one of the main processes in the brain. And then Nathalie and I went looking for what could be causative of this, and we found a link that was already described by Anke, which is the clearing mechanism of the brain. But we also find something new, and that was the involvement of the mitochondria. And the mitochondria are the powerhouse of the cell. They give us energy. And if there is an effect in the mitochondria, it could be that it's reflected in the energy metabolism. We found this in brain, but I wanted to show you because it's always nice and visual. We also took skin cells, and these are the skin cells of Cedric and the baby from your son. And we stained the mitochondria with a red color, and here you see the healthy individuals, and here you see the patients, and you see if we quantify the redness, the intensity is less, and it means that the mitochondria are less active. And my colleague, Dr. Nathalie van der Aa, she will elaborate on how this could be a possible treatment strategy and how she will tackle this in the clinic. This research is it's published in a very renowned and rewarded journal, and I really want to thank all the parents, and especially the parents from the family, that we were also able to publish on this. And I also want to highlight that between scientists, we were working very hard together. This was a very nice collaboration between Belgium, Israel with the Anke, Nathalie with all other labs. So may I please, it's not for me, but please for the donating family, ask a small applause because this is a prestigious donation that they made. But then I also want to point a last couple of things to the fore, and that's that there was a crowdfunding initiative on behalf of the German parent group to support ADNP research. And as I mentioned before, we raised over 30,000 euros for ADNP research. And then Frank and I were also very fortunate that with the same study that you donated, we also got a grant from the Marie de la Poix Foundation, also supporting our research. So thank you. And we use this money, of course, for drug screening investigations in ADNP-derived model systems. And today I would like to tell you how we tackle this in two different model systems. First, we took some blood of children, and then we go to the lab and we can make a cell line out of it. And we tested ketamine because, as you might know, ketamine is currently also an investigational drug for a syndrome. And ketamine, as you might or might not know, it's a drug that can induce anesthesia. So when you go to sleep before you get surgery, and it is also used in the party scene amongst young people, and it has some psychoactive effects. And there were indications in literature that below dose, so below the normal anesthetic dose, so subnarcotic, subanesthetic doses of ketamine, could turn on the ADNP gene and could increase again the content that was decreased, as I told you. So this was an assumption. So what did we do? We took the cell lines of 3 patients and 3 healthy individuals. We treated them with ketamine in a low dose and high dose for 40 minutes and for a longer time period. And we used also the Vunetides, the investigational drug of Villanagoses, as a control compound. And then we did some molecular work. And as I show you here, this is the therapeutic dose of ADNP. And we see that there is no increase in the ADNP levels. So the low dose ketamine does not increase ADNP protein levels. However, when we look at high dosing of ketamine, we see that it increases ADNP. That's very true. But at that concentration and time point, ketamine is toxic. We did the same experiment under the same conditions with the same cell lines with the Vunetide. And we use the Vunetide as a control because at these time points, we know that it doesn't work. It does work, but it doesn't increase ADNP. And we see that it is nicely stable. It doesn't change over time. Then we also wondered what are the processes in the cell that are affected by ketamine. And we have the healthy cell lines from the patients and the control individuals. They're not treated. And then we can clone them, and then we can treat the clones with ketamine. So we can, from the same individual, see in untreated and treated conditions what is happening. And we see that if we treat the cells with ketamine, that are essential processes deregulated that are already dysregulated by the ADNP mutation. So on top of the mutation, we see that ketamine is very detrimental for nervous system development, neuron formation, and differentiation. And we see also that it affects the skeleton of the cell. And this is, I pointed this out because I tested one marker for the cytoskeleton, and that is the tubulin. And we see that when we give low dose ketamine, that the tubulin content decreases. This is very detrimental because we need a very good skeleton of the cell, remember, in the mice for learning. And when we do this with the Vunetide, same experiment, we see that the whole cytoskeleton is not affected in expression. That's very nice. So we also use the money, of course, that we raised for to test the Vunetides. which is a small part of ADNP with neuroprotective properties. And this is in one ADNP mice in the lab, and we use it with a little plastic pipette tip, and we insert it in the nostrils of the mouse. And it's similar as Ilana's nasal spray, which she will talk later this day. And what we see early findings in our mouse model is that the unitide reinforces the cytoskeleton and it makes makes learning easier. But for this, to show you this in a better way, I was awarded a prize for science communication, and I was invited to make a video about my PhD topic. And some of you has already seen it on LinkedIn or Instagram, Facebook, or any type of platform, because I share it everywhere. But this video was actually made for today, and I would also like to take a moment to show, of course, this video. Born, you notice that some things, and it's always awkward to hear yourself. But I hope you enjoy it. Imagine you're expecting a baby. All tests indicate that you have a healthy pregnancy. However, when the baby is born, you notice that something's not quite right with its facial appearance. A few months later, doctors drop the bombshell. Your child might be suffering from a rare disease. Rare diseases often have genetic causes. For instance, a health model from the Kooy syndrome. Children affected by this syndrome present with severe autism, intellectual disability, and they struggle in daily life activities, such as speaking, walking, and learning. But what causes this complex condition? Well, mutations in a specific gene called ADNP, which is essential for the formation of the brain. But so far, there is no therapy to treat this syndrome. In a quest to treat the syndrome, we first need to have a better understanding of it. Therefore, I created a mouse model with a defect in the brain that closely resembles a child with the syndrome. In this way, I try to find differences between healthy and affected mouse brains to understand the abnormal mechanisms. We learned two things about the affected brain. First, the cells of the hippocampus are not fully developed. And this is important because the hippocampus is a brain region involved in memory and learning. Secondly, investigations to the structure of the brain cells, called the cytoskeleton, showed that it was not as strong as it should be. Fortunately, there is a ray of hope on the horizon, a special nasal spray. The spray contains a small part of the ADNP protein, and now I want to study the effect of this medicine in children with the health model from the Kooy syndrome. But for this, we first need to test it in our mouse model. And what do we see? The early findings in mice are extremely encouraging. It seems that this spray will help to restore ADNP levels in the brain and even make learning easier. So we hope that we will soon find the keys to unlock treatments for this disorder and also for many other rare diseases. So I hope that I could make myself a little bit clear on what this drug does and what it does in our mouse model, our Belgian mouse model. And I cannot show you the real results yet because they're extremely promising and we're working really hard on them, but I can tell you that we are going in the right direction. And then lastly, I have one more slide. And it's because I always want to ask something. And we are also continuing our quest and expanding our knowledge on the ADNP. And we are with Anke and with Kelly and with our whole team, we are doing a blood collection because we want to evaluate on the children and how they were like when they were young and how they are moving into their developmental trajectories. And we also therefore want to expand the clinical characterization. And we also want to investigate blood because we have a very nice research hypothesis within our lab. But for that, we need more blood or DNA. And secondly, I know many of you have already seen me made this announcement in the parent group on Facebook, but we are also looking into MRI images to have a better understanding of the brain during the development of the children. And therefore, I would really like to ask if we can have your report of the clinical geneticist, the MRI images on CD, disk, USB, pictures, everything that you have, and the report of the radiologist, because we are doing together with Anke also a clinical study on the brain of these children. So any information that you might want to have or share is really welcome. And then lastly, I want to thank the whole team here in Belgium that I work in, my boss Frank, our excellent interns that we have that I select. We have also amazing technicians in our laboratory, Ellen and Kevin, and they support us tremendously in this work. I want to thank the Research Foundation and especially the Research Foundation here from the parents in Germany that supported this work and make this nice presentation feasible for me. And here you see also one of the Belgian families when we were in the hospital to get blood. And then most of all, I would like to thank you guys, of course, for being here and for really bridging the way between patients and scientists. Thank you.
Work and for bringing this here today. I would like to ask you, are there any questions for Claudio? That's quite a few.
Hi, Thomas from Poland. First of all, thank you for a great talk. And I have a question about the divinitide. You mentioned that it improves cognitive features, right? And what about some physical features? I mean, if a child is severely impaired, doesn't walk, it doesn't sit independently, what about that?
For the clinical features, I would like that Ilana and the company that is here, that they would comment on this because they investigate this. I can only comment on the mouse work that we did. But in terms of the mice, we see that small milestones in terms of the opening of the eye after treatment, it's 1 day later. And we see this in males, Ilana sees it in females, but we see that the drug does something during the developmental trajectory.
Okay, thank you.
Okay. Are there any side effects by using the nasal spray in long term? No.
So the spray was tested in an FDA-approved clinical trial in patients with mild cognitive impairment, tauopathy, schizophrenia, and a bunch of other things, and it was safe and well tolerable.
Please raise your hand like a bit.
Hi, Stefan from Germany. Two questions. First, can you shortly tell us what hypo and hypermetallization is and how?
So we have the genetic codes, and I told you there's A and T and C and G. Methylation occurs most of the times at C, and you have to imagine the letter C and you put on a flag. When the flag is on the C, it means that less genes could be expressed. When you take away the flag, it means that more genes can be expressed. So hyper, more flags, less genes expressed. Hypo, take the flags away, more genes are expressed. This is in a very simple way, an explanation. It's more complex, of course.
Do you have this is email from the PO?
Yeah.
Hello.
Yeah.
I know there is a 3D model of the ADNP homebox protein in a database, internet database. Are there also 3D models of a dysfunctional ADNP protein?
Yes.
Do you have pictures from this and you know how the ADNP homebox protein looks like when it has and are there differences between the mutations?
Yes. So the 3D structure of ADNP is very big. It's like a telephone cord, if you remember the old telephones that you wired around. And when we have mutations, sometimes the telephone cord, it unfolds a little bit. And we need this like wiring because ADNP wraps around the DNA. So it needs to be wrapped around. And when you have a mutation, it can be that the wiring allows that it is less accessible to the DNA. And this is very important. And we are currently proving this in the lab, how this is working, how this is going. But in terms of 3D structural models, I've done it, Ilana has done it, and we see that there are differences.
The house you use as an adult brain or a newborn brain?
Sorry,
We can consider the brain of the mouse is adult brain now.
we have followed up on the brain during the development. So when they're newborn and when they're very like 10 weeks old adolescents. So we choose also the adolescent time because most of the children that are going to enroll in a clinical trial would be also in this critical period of time.
Any other questions?
At Verbruggen University with a mitochondrial dysfunction in ATP, but that was not found in other children, I assume. Now you're telling us there are problems in the mitochondrial ATP methylation, or somewhere
there.
Yeah, but can you tell me the problem of ADNP is that because mitochondria are very important for your brain cells, your muscle cells, your heart. Is that then probably a main problem? Or is it because the ADNP not works, you get mitochondrial problems?
We think it's because of ADNP doesn't work like that. It's a consequence. It's not. It's a consequence. But there are also arguments to turn this the other way around, because if you have the effect of mitochondria, and they happen before the mutation, it's like it keeps on rolling. But we especially control this with in the human brain with control, in the patient cell lines with the control, in the skin cells with the control. So we are pretty sure that this is a consequence of the ADNP mutation.
It's not a disease.
No,
No.
it could be mutation specific.
That's very true. We can treat it as a mitochondrial disease. We can give supplementation to boost the mitochondrial function, for example, Q10. And my colleague, Dr. Romera, she will tell you about the Q10 supplementation that she does in her practice.
Yes, thank you. At my home, you are known to me as the mouse man, so I'm honored to meet you at last. Very enthusiastic about how you present your story. At the end, for me as a parent, I see the holy grail. There's a nasal spray all of a sudden for me. Do we get any other information about the nasal spray today?
Yes. Okay. Yes. In the afternoon, there's the clinical trial team from the Unitide is here, and they will elaborate on how they will implement this spray.
Ah, okay.
That's the way. But what is the main problem? Is it infection? Is it epilepsy? Is it what kind of liver problems?
In particular, it was because the infection could not be controlled. The antibiotics were not working. Then he went into sepsis, and then he had multiple organ failure. So it was because the initial infection could not be controlled. But most of the children, it's controllable. They are in the emergency room, but it's under control. But this is something that is maybe informative to consult with your medical doctor when something's happening over there. But we are not quite sure why this is happening, because ADNP is almost not expressed in the liver. So it's a very intriguing case. But this is why organ donation is very important, because we need to learn more about this. Yes. Yes.
Thank you, everyone. Thank you, Claudio. I would like to invite everyone for coffee now. We'll have a short break. We're already running half an hour late. So let's make it a 20-minute break and come back at 11:45. 11:40. We will start at 11:40. Yeah.
Ja,
So this works. Can everybody hear me okay like this? Okay, so good morning, everyone. And first of all, thank you for the opportunity and invitation to speak here today. Today I will elaborate a bit on the research that we did on gait analysis in children with ADNP syndrome. So my name is Liz. I'm a physiotherapist by education, and I'm currently working as a researcher in the field of gait analysis in children and adolescents with several rare genetic diseases, amongst others ADNP syndrome. So let's first talk about walking. Walking is something we do. It's an automated task, and it's present in a lot of aspects in our daily life. It allows us to move around freely, both inside and outside of our houses. For example, to go to the kitchen, to walk to the kitchen and to do some basic household chores, but also, as we all did today, to come to Antwerp, walk into this room and join a social event like this. So basically, walking is an essential motor skill that allows us to be independent and mobile in daily life. But what is walking exactly? I bet that if I would ask you, how do you walk, that you could not give me a precise answer to that question. So to counter this in our field of gait analysis, we defined actually 5 prerequisites of normal gait. So if a person's walking pattern meets these 5 prerequisites,
we can say that it's a normal gait pattern and consequently would allow this person to be independent and mobile. So I'll take you through these 5 prerequisites really shortly. So basically, a gait pattern consists of 2 major phases. So we have the stance phase and the swing phase. During the stance phase, we need stability of our supporting leg to bear the weight of our body. Then, in the swing phase, we create propulsion and we swing our leg forward to create a step. So if the stance and swing phase is combined, we are able to put a step forward. If there's a good stance phase followed by a good swing phase, we have an adequate step length, so we can make put big steps forward. As walking is actually an infinite series of stance and swing phases combined, it's important to, after the swing phase, initiate the next stance phase, and we do this by pre-positioning our foot right for the next initial contact. So actually, if we have those 4 things, so the stance phase stability, the swing phase clearance, an adequate step length, and also a good pre-positioning for initial contact, we can say that the walking pattern is efficient and is normal. And then it takes us back to this slide. So if these 5 prerequisites of normal gait are met, it allows us to be independent and mobile in daily life. So that's really the gait pattern, the basics. So now we can apply this knowledge onto the ADNP syndrome. We know also from previous talks we had today that these children encounter motor developmental delay. This means that important motor milestones such as crawling, sitting, but also walking are achieved delayed during the development of the child. In ADNP syndrome specifically, the achievement of independent walking is delayed in around 86% of children. But next to these motor developmental delay, also other symptoms such as hypotonia or musculoskeletal abnormalities may impact these 5 prerequisites of normal gait, and thereby also the independency and the mobility of these children in daily life, which is of course a big problem. So that is what we actually, in our research, was the first thing we asked ourselves: do these children encounter difficulties with daily life mobility? To answer this research question, we used 2 questionnaires, so the Functional Mobility Scale and the Mobility Questionnaire 28. The Functional Mobility Scale is an observational performance measure to objectively measure how the child actually moves around in daily life. So the parent is asked to describe how the child moves around in 3 different conditions or distances. So in the home setting, on short distances, in the school setting, on middle-long distances, and then in the community setting, on longer distances of 500 meters and more. So eventually a child gets 3 scores, 1 on every distance, ranging from 1, being that a child who... Is there a pointer? So ranging from 1 to 6, being 1 that the child is wheelchair bound, and 6 being that the child is actually a fully independent walker and experiences no difficulties. If we then look at the mobility questionnaire, this is a questionnaire to objectively measure how much difficulty the child experiences during specific indoor and outdoor mobility activities. So here again, the parent is asked to score every activity on this questionnaire with a score ranging from 0 to 4. With 0, that the child experiences no difficulty at all with this activity, and 4, that the child is impossible to do the activity without help of another person. So let's then look at the results. So we questioned 13 children with an age ranging from 8 to 22 years old. And we asked the parents to fill in these questionnaires. So on this video, you can see the results of the functional mobility scale. So if you look at the bar on the left side here, these are the results for the functional mobility scale on the short distances. And it's actually striking to see that all the children achieve a score of 5 or 6, being that all children are independent walkers on these short distances. However, looking at the right-sided bar here, you can see a red section appearing this year. And that is a section of children that scored a score of 1 on this questionnaire. So being that they are wheelchair bound for these longer distances. This means that they cannot cover these long distances by foot. So this is already a first indication that something might be off with their gait pattern. If we then look further into detail into the mobility questionnaire, we see that children on several activities like running on grass, walking over obstacles, kicking a ball, or walking up and down the stairs experience significant difficulties. Even to that extent that almost 50% of children is not able to perform these activities without the help of any other person. So based on these 2 questionnaires, we can conclude that these children experience significant mobility difficulties during daily life, or at least a subset of children. So then the next question that rises is whether this could be related to these 5, to the problems in the gait pattern and to problems with these 5 prerequisites of normal gait. So that was our second research question. So what gait deviations are seen in children with ADNP syndrome, and might these be responsible for the mobility issues we see in these children? I think you can imagine that to answer this research question is a bit more through how we measure gait within our research. So we invited these same 13 children to our clinical gait lab in the Antwerp University Hospital, and we provided them of so-called retroflective markers. It are these small dots you can see here on the picture. And we put these markers on specific predefined places on the leg. And we just ask the child to walk up and down the hallway in our gait lab in the way they usually do during daily life. While walking, the movement of these markers is caught by special infrared cameras, and these can create a three-dimensional computerized version of the child walking, as you can see on the right side of this slide. So what is now the advantage of this method? This method actually allows us to get a detailed overview of all the movements occurring in the ankle, the knee, and the hip in all planes of motion while walking. It also allows us to compare this data to data on normal gait, and this way we can detect if there are any gait abnormalities present within any of these joints within a particular child. But this is a lot of information to process. So that's why in our research, we decided to also use the gait profile score. The gait profile score is actually one score representing the overall quality of the gait pattern. And it is calculated based on the movements occurring in the ankle, the knee, and the hip in 3 planes of motion while walking. It's not needed to understand the whole rationale behind this calculation, but the most important thing is that this score allows us to get one quick view of whether a child's gait pattern is impaired or normal. We use a cutoff score of 6 for normal gait, and the higher a person's GPS score is, the more deviant the pattern is from normal gait. So to conclude, we did not analyze this whole bunch of data at once, but we decided to first take a look at the gait profile score, and then to assess whether a child's gait pattern was normal or deviant. And then if there were deviations present, we further analyzed in what joints and in what planes of motion these deviations were situated. So then we can come to the results. So again, these same 13 children, and let's take a look at their gait profile scores. So in this figure, the gray box represents the cutoff score of 6 of normal gait. And you can see on the left side of this figure, the green bars that are located within or closely to this gray box, meaning that these children do not experience any difficulties, so we can consider these gait patterns as normal. However, on the right side, we see these orange and these red bars that are higher, these represent actually higher GPS scores. So in these children, we know, okay, we have to look deeper to see where the deviations are situated. So that's what we actually did during the analysis, and I will talk you through every joint and plane of motion. So where these deviations are situated now. So let's start off with the sagittal plane of motion. So the sagittal plane of motion is actually the plane of motion that divides our body into a left and a right side. And it's in this plane of motion that most movements occur during walking, because it consists of the flexion and extension movements of the ankle, the knee, and the hip. So in this figure again, you can see these green bars on the left, and then these higher orange and red bars on the right. So again, in the sagittal plane, there are some children not showing any deviations, but what we're most interested in is these red and orange bars. So we're going to see in what planes, in what joints these deviations are actually situated. Extend his hip as much as we wanted to see. So to conclude for the sagittal plane of motion, we saw some children who show normal movement patterns, but also some children with deviations both in the ankle, the knee, and the hip joint. And these deviations in these joints, they compromise a good foot positioning at initial contact, the stability during the stance phase, and also the clearance during the swim phase. So 2 more planes of motion to go. So let's continue to the coronal plane or the frontal plane of motion. And the frontal plane of motion actually runs like this, and it divides the body into a front and a back side, and it entails a sidewards movement during walking. What is striking if we look at this figure is that we don't see these red outlier bars. So in the frontal plane of motion, we did not see that much deviations as, for example, in the sagittal plane of motion. What we did see were some problems with the step width. In some children, we saw that the step width was wider than expected. So a normal step width is about 10 to 12 centimeters wide, but in some children, this was increased. An increased step width is actually a commonly used compensation for stability problems during walking, because the wider you put your feet apart, the broader your base of support is, and then it's more easy to counteract balance disturbances, and it's less likely to lose balance during gait. So this might also be related to the problems in the sagittal plane we saw with the knee. So for example, this could be a compensation that the children use to compensate for these stability problems. So for the frontal plane, we did not see that much issues, but we saw that there was an increased step width in some children that might be a compensation for some stability issues arising during the gait cycle. So the last plane is a transversal plane of motion, and this plane of motion divides the body into an upper and a lower part, and it contains rotational movements during walking. So to assess rotation during walking, we look actually at the parameter of foot progression angle, and the foot progression angle is actually the angle between the longitudinal axis of the foot and the line of progression during walking. In a normal gait pattern, it's about 10 degrees external rotation, but in some children we saw a more externally rotated foot progression angle, as you can see, of minus 25 degrees sometimes. So it's really the foot that were outward rotated. So what is the consequence of this outward rotated foot? The gait pattern gets actually less efficient, and the lever arm functions for the muscles that originate on the foot get also less favorable. So the gait pattern will get more tiring and less efficient when the foot is more externally rotated. So to conclude, for the transversal plane of motions, we again saw children with no deviations, but we also saw children with this externally rotated foot progression angle. So to come back onto our research question, so what gait deviations are present in children with ADNP syndrome? We can actually conclude that there are children who show no deviations, but there are also children who show deviations in all 3 planes of motions, and both in the ankle, the knee, and the hip joint. Now, what can we do about this? Unfortunately, that's a topic that was not covered in our research, and also not that much in research in general in this topic. but it would be really interesting to see how different treatment modalities would affect these gait deviations on a biomechanical point of view, but also to see if the functionality and the independence of these children gets better when we apply a therapy for this syndrome. But I cannot give you any clear answers on what to do with these gait deviations, but I think this research has already been a good first step to explore these things within these children. So what do I want you to remember of this presentation? I think that this slide gives a nice summary of what we did within our research and of the main things that we found. And yeah, I hope there will be some continuation, but that was my talk for now. Thank you.
Thank you, Liz, for your research on some of our patients. And I see Ilana and Claudio, you're going to have to fight for the mic.
I'm not British. I'm not Belgian or Italian. Liz, I just want to congratulate you on this nice talk, and I want to say that the feed patterns that you see, we see it also in mice. So it's a very nice correlation again between humans and mice. And now we'll pass on the mic because there is probably someone that wants to talk about how to correct it.
Not to correct. So Claudio's thesis in his line of last week of mine, we have like two more for the Harutyunyan ADNP syndrome, and we see it most definitely, and we also see a difference depending a little bit on the mutation. So if it's the child 719, we actually see more. So it might be nice to kind of try and correlate and see if there is a phenotype-genotype correlation, and we also knock down ADNP in the muscle, in the mice, and we see it's even more robust, and we can, the good news, we can correct it with the phenotype treatment. And what I wanted to ask is your methodology kind of EMA approved that could be carried to clinical trials? So I have a question.
So, if I understand your question correct, it is that if you ask if we could see these differences with this methodology, or
can implement if this would be approved by regulatory authorities.
If we could use this to evaluate treatments.
Yeah.
Yeah, I think it's a really interesting point of view to because these children encounter a whole bunch of symptoms, and I think it's important to, if we get a possible solution for the disease, to also see to get evaluation techniques that can assess whether this therapy might work, but on all these aspects of the symptoms these children experience. So I think it's not only this, but we need evaluation tools that are sensitive enough to test this whole area of symptoms that these children encounter, of which gait analysis might be one of them. Yeah.
Okay, we'll have time for one more question. Sorry.
I will try. Thank you very much, Mauricio. Thank you very much for your talk. I think it's tremendously helpful for a parent of an ADNP child, because we always get questions of doctors. It's a bit unclear to us why she doesn't walk, so she doesn't like really walking, specifically, as you mentioned, outside of the house or on specific surfaces. So I have actually two questions. So first of all, would it still be possible for children to take part of the gait analysis? And secondly, we have a sort of solution now from the physiotherapist to have insoles in the shoes. Do you think this would be beneficial, or at least not hurt her, considering your expertise? So thank you very much again.
So I think the first question, this study is already finished up, but we can talk about it later just to see if there are maybe opportunities. And I think for the question about the therapy, I think now the therapy to counteract these gait deviations is really individualized, so also based on individual 3D gait analysis. So, and I think insoles or orthoses might be really helpful to really work on these gait deviations. But it would be even more interesting to see if a cure for the disease would also have an impact on these gait abnormalities, because it might be that it's just something that has to do with musculoskeletal structures during the development of the child, and then an insole would be a better solution than I would imagine a nose spray would be. So yeah, it's also a thing of really implementing an early intervention for walking as well. That's important for these children. Yeah.
Okay, I'll take a last question.
No, it's more, we don't know exactly where this, I cannot say that the problem with the knee is due to hypotonia. It's more like every child has its own set of symptoms. And I think this all contributes to the problems in gait we see. But I think it's difficult to pinpoint one specific problem that causes these gait problems. I think it's a story of N and not one specific.
Because our daughter is stretching her knee like in a boat. It's not normal. Also, she has scoliosis and lordose, but is it all the same because of hypotonia?
I think it's all related, and I think the main thing is also these developmental delays these children experience. So because of the delays because the development goes more slowly, it's also more difficult for these children to learn how to walk. And I think they also learn themselves a pattern that works for them. And sometimes it's also good that they found compensation strategies for their own to be able to walk. So it's not that we have to fix all these gait problems we see. Sometimes it's a functional adaptations these children have made. And so it's, I think it's a combination of several factors that contribute to these gait deviations. And it's especially not always bad that these deviations happen, but it's interesting to see if there are maybe similarities within these children, or indeed, as you said, between different mutations. So I think there's a lot of research to be done in this field, and we're not there yet.
We're not there yet. Thank you, Liz. And for the next talk, I'd like to invite Kelly, who everybody knows very well, Kelly Verbruggen, together with Anke van Dijck and Lusine Harutyunyan, who've been doing some work on the therapeutic support trajectory.
Okay, thank you, Nathalie, for the introduction. So we are really happy to introduce to you this topic, actually, during this very interesting day already. But I honestly believe that what we'll show now is of a big importance to all of us. And why is that? Because I really do believe that this care path that Lusine will explain about will give us some guidance and some support in our way on how to deal and handle with our children. So it will give guidance and support, but also, and then in a later stage, Nathalie van der Aa will explain on supplements to support our children. So this care path has been written together with the Antwerp team, and we made a poster. Is it here somewhere?
During the lunch break, it will be.
Great. Okay, so we made a poster of the care path. And we really want to have your feedback on it. Are we still missing elements in it? And then we can add it and we can change it and we can publish it. So that would be a very great step forward, I think. So please welcome Lusine to the floor. Thank you.
Okay. Welcome, everyone. I have
heard a lot about many of you, but I think you don't know me. I am Lusine Harutyunyan. I am a PhD student under supervision of Dr. Anke van Dijck. We are in the research group called Disability Studies under the general research group of Family Medicine and Population Health. As Claudio is more of a molecular guy, I'm more of a clinical girl. I started a few months ago, and I'm doing research on the clinical picture and the development of Hellsworth van der Aas syndrome. I was looking from the back of the chapel to the group, and it's actually quite remarkable how many different types of people are present here today, from fundamental researchers to clinicians to parents, and with the sole purpose of learning more about the syndrome, and also to share experiences and seek support from each other, which is very inspiring and beautiful. So we all know that Hellsworth van der Aas syndrome requires a multidisciplinary approach to care. After initial diagnosis, a comprehensive evaluation system is recommended to address various concerns and provide appropriate interventions. So we attempted at a first version of a care pathway to encompass all and give you all the evaluation systems, at least in Belgium, and the different support systems available for our ADNP children, if I can call it like that. I first wanted to start off with this very cute picture of Cedric and Mick, who are the kids of Kelly and Mark, and Mick is the son of Gaby, who is here as well. I wanted to start off with this picture just to show that Hellsworth van der Aas syndrome has a face, and these are real people, and refocus to why we are doing what we are doing. So we came up with 5 main phases. From pre-care phase to diagnostic phase, treatment phase, short-term follow-up, and long-term follow-up. And I'll start with the pre-care phase, which starts when the first symptoms present itself in the children. Some of the first symptoms that are usually noticed at a very young age are hypotonia, any severe speech and motor delay, changes in behavior, signs of autism, mild to severe intellectual disability. Any facial features that are very noticeable are a big forehead, small nose, large eyebrows that are usually present in the children. It is possible to start early therapy even before diagnosis is made, because as Liz said before, early therapy, for example, physiotherapy can be can help tremendously with the development of the child, but also speech therapy, behavioral therapy, and sensory therapy. After the first symptoms are noticed, the first referral happens, and that triggers the diagnostic phase. The first discussions about diagnosis happens between the genetics and the neurology department, where blood collection is performed for DNA testing. After the result is known, the second discussion is initiated between different disciplines that include cardiology, urology, and nephrology. After the diagnosis is made, the start of the treatment planning can be initiated. This is in Belgium. The children and the parents are referred to the Center of Developmental Disorders, or Centrum voor Ontwikkelingsstoornissen, that is present in different university hospitals across Belgium. I know in Antwerp it is for children that are under the age of 5, and I searched it up in Leuven, it is under the age of 3. It is important to know these centers are not for treatment. They do different developmental and behavioral testing, and based on these tests and these results, they give advice to the patients, to the children and the parents. The next step is a guidance conversation that should be conducted ideally by the physician. At first, that gives parents the initial tools to find further financial and social help, and will be followed up by the social worker and the case manager or psychologist later in the trajectory. The Center of Developmental Disorders and the guidance conversation are necessary to identify the needs of the parents and the children. A next step is psychosocial support in the treatment planning. This can serve as a starting point for referral to governmental agencies, such as increased care allowance or a personal assistance budget. That is the case in Belgium. And then another important part in the treatment planning is the ADNP multidisciplinary consult that, as the name itself says, includes many different specializations to, at an early stage of the diagnosis, encompass the full clinical picture of the child and make sure that everything is noticed and nothing is overlooked. In Belgium, the multidisciplinary consult consists of the pediatric neurologist and the geneticist. The next step is the actual treatment phase for the different manifestations of Hels-Mortel van der Aa syndrome. This can go from nutritional support to treatment for constipation and reflux, but also treatment of sleep disorders, etc. The treatment plan should undergo systematic assessment to preserve the quality of life of the child and the parents, but also to evaluate any psychosocial and rehabilitation needs. This should be conducted in a structural way in the context of an additional multidisciplinary consult to cover the full clinical picture of the patients. And as the disease is a developmental disease, it can progress over time, and that assessment is very crucial. Any adjustment of treatment as a consequence of the change in clinical picture over time depends on the age and the development, of course. So some manifestations can improve or even worsen on a later age as they grow up. Next, the medical support, the social and financial support are also very crucial, not only to the child, but also to the parents. In Belgium, there are several governmental agencies where you can seek help, such as the agency in Belgium for increased child benefits, but also the VAPH, or the Vlaams Agentschap voor Personen met Handicap, to get your disability card and aid applications and to apply for personal assistance budgets. There's also different types of organizations such as Kind en Gezin and the CLB that will help you in search for the right school for your child. And then there's the short-term follow-up. The active involvement of the MDC, so the multidisciplinary consult, stays very important. It is key for effective treatment plan and the detection of any change in the clinical picture, and also early education of parents by caregivers regarding treatment options, financial support from government agencies, and the presence of any patient organizations, different options for sports and day activities. So ideally, we'd hope that the MDC is fixed protocol that is applied in all countries, that where patients that are first diagnosed or children that are first diagnosed get referred to and get the right help and support. A case manager can be appointed as a short-term follow-up, but also in the long-term follow-up, as I will tell you more later. He follows up on all different medical consults, but also on any functional and lifestyle medicine, about which Nathalie will tell you more about after my presentation. The case manager composes and implements an integrated treatment plan to assure appropriate care for the patient. He plans and coordinates healthcare services. And then next to that, support, family and community support is also very important. It can be the parents, but also formal and informal caregivers. And then in the community, there are organizations such as Verbruggen, where you can find guidance for children with intellectual disabilities. A social worker can help follow up on all governmental agencies, as it can be quite a lot, and many things that you didn't hear of, but are where you are eligible for. So that's a social worker that can help you with that. When the diagnosis is made at a young age, there are crucial questions to be asked about school and daycare activities. Is the child going to school? Yes or no? If it is, what type of education is going to follow? If it's not, what are the alternatives? Are they included in any hobbies or G sports? Belgium and the Netherlands are leading examples in this aspect. They have very good and very well worked out G sport organizations and groups where the children can be a part of. And then also AKB is a youth movement that I heard from Kelly yesterday does not have any age restrictions, which is, I think, very beautiful and makes sense. So for the long-term follow-up, again, I keep repeating myself, the MDC stays important for regular evaluations, continuous monitoring of the developmental process and any medical conditions. They adjust treatments and support strategies as the individual grows and their needs change. Not only developments in the clinical picture, but also developments in the research regarding to ADNP and the syndrome are something that the case manager needs to follow up on to make the family aware of any changes. Another important thing is the transitioning of children with Hels-Moortel van der Aa syndrome from pediatric medicine to adult medicine. That transition can be actually very stressful, I can imagine, since the family and the children have created a relationship with the healthcare workers in the pediatric department. Once they turn 16 or 18 years old, they are kind of forced to transition to the adult medicine department that has other doctors, other nurses. They have a different way of working, and they include other specializations. So we want to work on a policy that helps the children, the parents, to make a smooth transitioning. And we are of the meaning that this should be exceed age restrictions, and that there's implementation of a transition phase where the needs of the patients are continuously met. And then to end it off, family and community support stays essential during this phase, especially as the kid grows up. It will need patient empowerment, also in schools and the kindergarten. It's essential that the kid stays active in any way possible in hobbies and G-sport, and to ensure that there's community integration and inclusion within society, whatever that means for you. So, like Kelly said, since we highly value the input of all the stakeholders, there will be a poster and some post-its and a pen besides it. Write your ideas, suggestions, feedback on it, and paste it on the poster wherever you find fit. The development of this care pathway was made possible by my supervisor, Dr. Anke van Dijck, Nathalie van der Aa, and Kelly, of course, who is also always so kind to share her expertise and her experience with us. I want to close off with some personal remarks. So my journey in the ADNP and Hels-Moortel van der Aa syndrome world started only a few months ago. And even though I hadn't met many of you before today, I learned a lot about our children from, if I can say it already, from the things my colleagues have told me about. The topic is all about the patients. Usually, as researchers, we often, I think Claudio knows, we often immerse ourselves in numbers and data, and sometimes we lose sight of the fact that these statistics represent real people with real experiences, with sleepless nights and a feeling of helplessness and challenges that defy control. So as caregivers, and most importantly as parents, your unwavering dedication and courage to secure the best care for your children is nothing short of remarkable. I am deeply moved by your strength and resilience. I just want to close off by reassuring, and I think I can say this from our whole group, by reassuring you that all the research we do is always holding you guys in our thoughts. We acknowledge that it is not easy, But together we persevere in our pursuit of therapies for your children, for our children. I want to express my gratitude, and it was truly an honor to stand here in front of you and present today.
Very much, Lusine, for these beautiful words and for sharing the trajectory path that Kelly has mainly started to bring together. Does anyone has any sharing or any question about this for either Lusine or Kelly?
Yes, thank you for sharing this. I think it's a very wonderful overview of things what can help us. The only thing I was just wondering, looking at our own son, is that the behavioral disability is limiting a lot of potentials. And that might be something to bear in mind, because finding additional support is sometimes very difficult due to the behavioral issues. So it's a great work. Thanks.
Yeah, well, René, we know it is so different for all of our children, but within the care path, we want to take into account all possibilities. And we want to stress the fact that, yeah, I know, like for my son, he's quite mild within the syndrome, and he can really do a lot of stuff. But so, yeah, I think it's just important that everyone knows that because Cedric, he had already at the age of, I think it was 8 months, that he started with therapy. And I do have a strong belief that the early start of it, that it has big influence on it. So, which of course doesn't mean that for other children who are more severe, thing in this ADNP, it is something completely different. I do understand that the most important thing I wanted to stress into this care path is actually the fact that for all this family support, all these governmental organizations, we need to find it for ourselves. There is no one who is explaining to you where you have to go, what you have to look for. And I think that is very important to know from the start, and that you will be helped from the start. Because Cedric is almost 16, and I just got my personal assistant budget. And it is all of us are so hard fighting, and we always stay fighting for our kids, but it is taking so much energy that it is really hard, and therefore we thought this care path will be of a great help, although it will not be suitable to every one of us. Okay, thank you, René.
Yeah, for me it's likewise. We have also big behavioral problems, and we see it a lot also on the Facebook group. And the care path maybe can help us to indicate which medicine maybe we can try, or which direction we need to go to keep them a little bit calm and to function in day, because the problems are really, really big. It's not a little problem. And maybe we can also do more research to that, because I think that there is not enough research done about that yet. And that's the main problem in day by us.
Yeah, and therefore I think, Marita Elisa, yeah, therefore I think also a care path, it should not only be for Hils-Moortel van der Aa syndrome. Because to be honest, when we look at Down syndrome, because a few of the friends of Cedric have Down syndrome, and when you look to, for sure they have a totally different clinical approach, but next to that, the care path of all of the support and the behavioral issues and all these type of things, it's quite the same. So, and I think we should more unite with different patient communities, instead of only focusing on ours maybe, but that is, yeah, still a lot of work to do.
Any other questions or remarks, Kelly, or we move on to the next speaker, which is me. Sorry.
I think Gaby has a question.
Oh, sorry. Gaby, I didn't see that. Sorry. Thank you, Lusine.
It's a good thing. I also thought about why is it not a system like that, like a care path. So I think it will be very positive thing. Thank you.
Thank you, Gaby. Thank you.
So indeed, now we have our next speaker, Nathalie who will discuss a bit more on the functional and lifestyle medicine, of which I truly believe which can have a good impact on the daily lives of our children.
Thank you, Kelly. So some of you may know that shortly after the syndrome was published, I left the University Hospital and the University of Antwerp to pursue on another mission as a general pediatrician again. And I've always been intrigued with children with these rare developmental disorders and severe behavioral problems, psychiatric features like autism or attention deficit or other problems, how we could help them with other things than psychopharmaca, because psychiatric drugs are used a lot in behavioral problems and in psychiatric disorders related to syndromes. And we see a lot of differences in the responses of the patients that cannot really be explained. So going back to general pediatrics, I had quite a lot of people referred to me by other pediatricians that had emotional or psychiatric problems with or without syndromic features. And I sort of took the path of looking into complementary medicine, which is not something that is done a lot in Belgium. In America, the functional medicine has been developed for 20 years on that path. In Belgium, the complementary therapies and also molecular medicine are more in therapists. Some osteopaths, dietitians do lifestyle medicine. Not a lot of doctors are involved with that. But for me, it was very interesting to look at it from the world where I came from, so inspired by the science, to look at what was known by experience in lifestyle. So, and that's what got me onto this integrative medicine, which is actually a combination of conventional therapy. So I do see, I do use a lot of drugs in integrative medicine, but I also use complementary and alternative therapies. I do this in a highly personalized manner. So I look patient per patient to individualize and tailor supportive complementary therapy to meet the unique needs of the patient. And as you see by these three circles here, it's gone. Here it is. So there is an overlap between what the genetics of the patient is, the epigenetics of the patient is. Epigenetics is actually about those flags that Claudio was talking about. It's about whether the gene is expressed or not, whether the gene is on or off at one certain moment, because genes do get turned on and off during the lifestyle. For instance, once we're an adult, there are certain genes that we don't need anymore to grow into an adult, so those get turned off. So, and also environment they all play a role in what we see in the clinics of the patient. So there is always, even if you have an ADNP mutation, there's always this small gap where you can still do something for the client or for the patient through working on epigenetics, so on the flags, on the methylation, or working on lifestyle factors such as diet or other things. So it also entails a holistic approach where we look not only at the body, but also at the mental aspect of the patient, but also at emotions and spiritual aspects of the human being, actually. So mainly on addressing environmental and modifiable lifestyle factors, there has been a lot of focus by functional medicine therapists on exposure of to environmental toxins. There has been a focus on dietary measures and the use of targeted dietary supplements. There's also a lot written on the microbiome. I'll come back to that. There has been some interest in what exercise can do for you, what sleep can do for you, and other various interventions that can help to improve your environment and hence your disease or your well-being. So what is remarkable, or what I see in the Hels-Moortel van der Aa syndrome patients, that there is quite a few differences in the level of cognition. There is quite a few differences in the level of behavioral problems. I know a lot of patients do suffer with severe behavioral problems, as in many of these syndromes, but there are differences. And also there is a lot of differences in what we call comorbidities, which is other health issues that present in these patient groups. And that can be problems with the immune system, as we talked about, with the stomach and the gut and the intestines. There's a lot of feeding problems. There's a lot of problems of constipation or diarrhea. There's also some allergies and sensitivities to specific food. And then the mitochondrial dysfunction that now has been so nicely in vitro being demonstrated by Claudio, that there is mitochondrial dysfunction. And coming back to the mitochondria, it's that powerhouse. It's the part of the cell that takes care of our energy. And a cell cannot work without energy. And in the last few years, a lot has been known in different labs that study mitochondria about the importance of the mitochondrial function in cognition, in learning, in muscle tone, in using muscles, but also in even in prevention of cancer, in anti-aging. So the mitochondrion really is a key to a lot of things. Also, we see sometimes problems of what we call poor detoxification. This means how do we get rid of toxics of the body? And then inflammation is what is everything that's got to do with infection, but not only infection, also autoimmune inflammation. So on the other hand, as we learned from Claudio today, and as we learned in the last 2 days from a lot of scientists all over the world, there is an increasing knowledge and increasing insights on the function of ADNP and the impact on the different organs. Now, next to that, there is also an increasing understanding how neurodevelopmental and behavioral outcome is influenced by both genetics and other factors. So this draft is maybe a bit complex, but it was published this year, and it tells us a lot on how everything in our environment that we've been exposed to, even in generations before, can have an impact on our gut and on the bacteria living in our gut, and how this has an impact on the genome. And the genome is much bigger than only ADNP, and we know that there is, from the whole genome, there is interactions with ADNP, and we know also that ADNP plays a role in other genes as well. So the whole genome is important for the clinical, for the clinical aspects, for the severity of, for instance, your behavioral issues, or the severity of other symptoms in Helms-Morsel-Van syndrome, or other syndromes for that matter. So we are going to, I was looked at what environmental factors have been studied that I can modify. So I've been looking at the accumulation of heavy metals, which has always been a big issue in autism, in pediatrics, about the diet and the use of specific dietary measures. I've looked at the gut microbiota, and there's also some literature even on how exposure. Do you want me to take the other microphone? Oh, okay, I'm sorry. I'll put it here. Yeah. I'm sorry. Anyway, there's some literature also on electromagnetic fields and also on other therapeutic interventions like osteopathy, occupational therapy, speech therapy, and so on. So this is also a part of complementary medicine. Anyway, I got a lot of questions regarding toxins and specifically heavy metals. I sometimes get people in my clinic that want blood tests for what heavy metals play a role. And I've been looking into the relationship between heavy metals and autism, and there is some literature on two heavy metals. One is aluminium, of course, that's also now related to Alzheimer's. And the other one is, I can't think of it. Mercury, yeah, from the fish. I was just seeing all these fish. Mercury and aluminium are two heavy metals that have been discussed in the literature as an environmental factor that can worsen autism. So there is some evidence for detoxing on these heavy metals. If you may have heard about this, there is some evidence, but there's not that many studies. But I know, like in the States, a lot of children with autism and behavioral disorders get detoxifying protocols with zeolite or fulvic acid, something else. I myself, I use spirulina, as it's very harmless and contributes to other aspects of improvement of cell metabolism. On the impact of nutrition has been published since 1966. The first reports were in schizophrenia, where after eliminating gluten out of the diet, there was seen that there was a huge improvement in the symptoms. So, and there have been lots of subsequent studies on the effects of gluten and casein, which is the protein that's in cow's milk. And there has been a lot of explanation about why that would be, and it also seems to come down to what's happening in the gut and how the signaling from the gut to the brain is harmed by casomorphins and glutamorphins. So just to show to you that in the last, say, 10 years, there's been such an increase in the publication of papers on the impact of nutrition in autism. It's amazing. I didn't read them all, but I wrote this good review that's taking all of them together. And as you can see, there is some reporting on behavioral improvement in some of these studies, in these 4 studies with follow-up from 12 months to 4 years. So I'm not going to discuss them all. There is also some that have non-significant improve in behavioral problems or in some blood sample results on metabolites. So it is thought that nutrition, the improvement that is seen on behavior through nutrition has to do with the microbiome in the gut. And in the last 10 years, I think the microbiome is the most published thing in general. It pops up in everything. And from 2014, the papers on the microbiome and the role of the microbiome in autism have been really, really big. So the microbiome, what is it? It's the collection of all the microbes that live in our intestine. And they are very important because they digest our food, they train our immune system, they prevent harmful bacteria to come into the gut. And also they produce all kinds of elements that make signals to the brain, and that means that it affects the brain health. So GABA, for instance, for people that have heard of it, which is a neurotransmitter that causes relaxation, is produced by some strains of bacteria in the gut. And we know for sure that from studies that it is very different in children with autism. So there is a lower diversity and a lower amount of good strains in children with autism. And I think, Ilana, that you've also shown that the mouse microbiome in ADNP mice is completely different from the bacteria in the gut of healthy mice. So, and also we see that autism is a lot associated with problems of gut and intestines. For instance, in our syndrome, we see constipation quite a lot. And this has also to do with the fact that there is different strains of bacteria in the bowel and in the gut of the children. I'm just going to skip this slide. So how can we impact this microbiome? We know for sure that what the mother has living in her gut plays a very important role when she's pregnant and when the child is born. Also, if the child is delivered through cesarean section, for instance, it cannot inhale bacteria from the mother by passing through the birth canal. So that is something that we now try to work on in pregnant women on a good microbiome. We can also impact, of course, the microbiome by antibiotics. Certain antibiotics will make sure the bad strains go, but there is also a lot of microbiotics, antibiotics that are used, for instance, in airway infections that get rid of the good microbiome as well. So it's important always to know how many antibiotics a child has had in their young lives, because that might impact also how the behavior develops. Nutrition, of course, has an impact on the microbiome, and also if the gut cells that produce the mucus where those bacteria live is good, the gut lining, if that works well, because we do change our gut cells every 4 to 5 days, and we can have an impact by feeding them the right vitamins and minerals and amino acids. So you get a good enterocyte or a gut cell that produces the mucus where the good bacteria like to live. And then another way to impact the microbiome, and this is something that's been in the last few years has been used in several diseases that are associated with microbiotic issues, are fecal microbiota transplantation, or FMT. And there have been some studies that have that have published some good results on an improvement, a substantial improvement actually, on behavioral issues as well as all the gastrointestinal symptoms. So, and there's also been a lot of tests after transplantations where blood sampling of certain metabolites that are important for the brain are more similar to normally developing children after a fecal microbiotic transplant. So I also get a lot of questions on what can we give our child to improve vitamin, mineral, and supplement-wise. Now, of course, in children with autism, there is a susceptibility to nutritional deficiencies because often these children self-restrict in good nutrition. And also, I think in our modern life, a lot of sorry, I lost the word in English. A lot of food doesn't contain enough nutrition anymore. So we do get nutritional deficiencies in a lot of children, and then also especially in autism. And these children do need even more vitamins and minerals than other children, because sometimes they are on drugs for gastrointestinal reflux, the reflex medications that are lowering stomach acid. Stomach acid, of course, has the function to make sure that the food is digested and all the elements that are needed for the body can come out of the food, where if you take these medications, you miss a lot. For instance, magnesium is a big one that is not released when there is stomach acid reduction in reflux patients. Also, there's other drug-induced nutrient deficiencies. Anti-epileptic drugs can cause nutrient deficiencies, vitamin deficiencies, other psychiatric drugs as well. So, and besides of that, also we see, as Claudio said, all these flags, these methylation problems, that these children need more vitamins to support the methylation process. And the vitamins that support that process are the B vitamins, B12, B9, B6. And also these children have sometimes, as ADNP interacts with a lot of other genes, may have some metabolic problems, so problems in the metabolization, so in the metabolization. In metabolism, yeah, in metabolism. So in metabolism, together with the mitochondrial problems, so with the energy supply, and also there has been some papers on antibodies to syrup. So this is an immune problem to proteins and transporters that act in the brain. So there have been some vitamins and minerals have been studied in these children, and there have been studies on an improvement in sleep problems, mainly that was seen, but also in all the GI symptoms. The main, I think the main contribution of the vitamins are an improvement in the energy of the cell, so the cell can function better. And that could be any cell going from the stomach to the brain. This is, I just added this table of a study that was done in 2011 in America with a supplement in a large cohort of children with autism. And the blue study is a study that was done recently. The orange study is done by the same group in 2011. It was repeated recently. And the gray blocks is the placebo. So this was a placebo-controlled randomized study. And they saw in autism-related symptoms a large improvement compared to the placebo group, as in language and comprehension, as in speech development, sociability. There's a smaller difference. This is with regards to tantrums, whereas the placebo group seemed to have got worse tantrums compared to the other groups. And this is hyperactivity also got worse in the placebo group, but not in the group that got, this is a multivitamin mineral supplement. Omega-3 fatty acid, some of you may have heard of that, has also been studied, which is an important constituent of all cells and has a high level of all membranes of cell, has a high level in the brain matter, brain cells, and protects against oxidative stress. And oxidative stress is actually self-harm to the body, in short. So it also has an effect on the gut, and it's a deficiency in DHA omega-3 has been linked to neurodevelopmental problems in general. Other nutrients that have been studied are carnitine, coenzyme Q10, L-theanine, and phosphatidylserine, which are also working on Q10 and carnitine. Q10 and carnitine are working on mainly on the mitochondria, and L-theanine and the other one are working on the receptors to bring rest to the nervous system. So if I look at these studies, I see that interventions in lifestyle and environment have demonstrated some promising improvements. There is always, if we look clearly, there's always a possible degree of expectation bias in every study. But there have been well-documented cases of recovery with intervening on a lot of environmental factors, such as diet, with supplements, with antimicrobials, with speech support, with occupational therapy, osteopathy, and so on. I personalize this to the child's clinical presentation, labs, tests, and the need of the situation. And there's been a recent remarkable paper on how parents just made a personalized lifestyle for their twin girls that were born premature, and that had an autism diagnosis at the age of 15 months with quite high ATEC scores, which are scores for the therapy for autism. And they were involved with a pediatrician that set up a personalized therapeutic functional medicine plan of craniosacral therapy, speech therapy, but also looked at blood results, at gut inflammation, chemical toxicants, and they had a really, really good response to a combination of all these modifications. So there is some evidence, and these therapies can even be combined with drugs that target the problem or that target the gene, I'm sure. And it might be interesting to set up a pilot study for evaluating the use of a well-chosen supplement in our patient group. So, and last but not least, Daniel Siegel is an American Californian pediatrician and psychiatrist who inspired me very much in my doctor-patient relationship by his discussion of what we call interpersonal neurobiology and how interpersonal experience shapes the minds as it continues to develop throughout the lifespan. And interactions with the environment, especially relationships with other people, directly shape the development of the brain structure and function. And I don't want to mention this solely because of the patient group, but especially because of the parental gathering that we have here and how we can all help our brains develop and are thinking about the syndrome. Thank you.
Thank you, Nathalie, for the inspiring talk. We are just before lunch, so maybe 2, maximum 3 questions for Nathalie.
Because there are a lot of stands.
Yeah, I use it depends on the age of the child. It depends on the symptoms, of course, and on the food. It depends a lot on the nutrition of the child as well, because if you use probiotics, you want them to stay there and to grow there. So nutrition is very important, as this gives the fiber to the right stems. Bifidobacteria have been definitely have been shown to have a big impact on as they are responsible of GABA production on the behavior. So bifidobacteria, lactobacillus, those are the 2 main ones. Mm. Yeah, it's not that easy. It's not that easy because what happens to, I mean, if there is another bacteria in there, have you had a microbiome testing in your child? No, in the feces. No. So it also depends on what other stems are present and how quickly they grow for the lactobacillus and the bifidobacillus. I don't do it regularly, but for me, I'm more working on experience, and I add supplements to improve the mucus production of the enterocytes. Yeah.
Yeah.
Yeah. It's also important how the supplement has been developed, what substance, because yeah.
A long time ago, good study to supplements, but it's very difficult because you don't know if it works or too much, is it too less? Yeah. So it's very interesting.
Yeah. Okay, more questions?
Excuse me, I don't hear you.
She will ask a question in French, and I will try to translate.
Okay, thank you. Thank you.
They have done the transplantation of fecal matter
All right.
on a Howard.
All right. When you were 6 years old, you had a fecal microbiome transplant. Mm-hmm.
It's only available in Belgium when they have that.
That's species of Clostridium. And then first they give the...
It's only available for children that have Clostridium difficile, who is a bacteria, bad bacteria that you can get in the gut. And it was because we... So we used this canal to ask transplantation. But so you told us about TMF could be a good idea, but
No, there's been some studies on it, because I haven't seen any patients, but it's interesting to hear your idea. C'est intéressant que vous expliquiez comment c'est votre expérience avec ça.
Oui, et donc on a fait les tests de développement neurologique avant et après.
Ouais.
Et on voit vraiment une différence. Et à l'école, il s'est mis à apprendre à lire, il faisait toutes des choses qu'il ne faisait pas. Son langage s'est vraiment amélioré dans ses mouvements. Donc, c'est surtout au niveau du langage qu'il y a eu un gros changement.
Oui.
Et de la concentration aussi. Et forcément, comme on aime toujours bien faire les tests avant-après, tous ces tests au niveau des intestins aussi montrent l'amélioration. Comme il était incapable d'avoir justement des lactobacilles, même par supplément de probiotiques, ça ne marchait pas. Et avec la transplantation fécale, c'est parti, c'est développé. Et je voulais juste dire que apparemment, à Gand, il existe une banque fécale à l'hôpital de Gand. Et donc là où je veux en venir, c'est finalement, est-ce qu'il n'y a pas moyen d'obtenir pour les patients ce qu'on appelle des gélules fécales?
Oui,
Voilà.
oui, ça, on doit rechercher, c'est possible. Hein? Just to translate for a minute, this is the mother of Howard who had a fecal transplant here in Belgium 2 years ago, and who's had developmental testing before and after, and there's been quite an improvement. Her question to me is, it would be interesting to continue to use capsules with the fecal transplant strains that help him, because before she wasn't able to get the lactobacillus in the gut of her child, and the transplant has helped to, if I say it correctly, to help with that. So yeah, these are all, I just looked into the literature. There's so many great ideas to look into, but I surely want to be involved and we could contact Ghent on that question.
Et la deuxième chose que je voulais dire, c'est qu'avant de faire ça, même si on a fait toutes les techniques naturelles et ce genre de choses, on fait quand même un grand nettoyage, entre guillemets, quand même on prend un antibiotique comme l'azithromycine. Parce qu'en fait, ça change tout. Ça marche mieux si on fait l'azithromycine, un lavement, et puis directement, on donne la transplantation fécale.
Oui.
Et je veux dire que quand il avait 3 ans, on donnait
3 ans. That's 3 years ago, and they give antibiotics before they.
Quand il avait 3 ans, et on avait déjà fait tous des tests au niveau du microbiome, et donc on s'est dit, mais qu'est-ce qu'on peut faire? Il a tellement des parasites, des mauvaises bactéries, des toxines, on ne comprenait rien. On s'est dit, on va le prendre, on va le jeter à la poubelle. Et en fait, à un moment donné, on a dit, OK, on va prendre l'azithromycine. Non, le métronidazole, on avait fait. Et là, on avait donné une dose pendant 2 semaines. Donc, c'était quand il avait 3 ans. Pendant 2 semaines par suppositoire. Et là, on a vraiment vu des changements qui étaient vraiment spectaculaires. Donc, notamment au niveau des pieds, par exemple. Il marchait comme un pingouin, les pieds écartés. Tous ses pieds sont devenus normaux. Et à l'époque, je devais écrire le nombre de mots qu'il disait. Il disait 30 mots et en 2 semaines, il était passé de 30 mots à 300 mots. Oui. Et donc, après ce métronidazole qu'on avait donné, oui, on a directement donné un probiotique qui s'appelle Vivomix 400. Parce qu'ils le font en 100 et en 400. Et donc, ça, je veux dire que ça, par exemple, tout le monde pourrait essayer chez soi. Quoi. Je voulais quand même juste donner ce genre d'informations.
Très intéressant, merci. Oui. So she said that before she started on the probiotics, her child had, at the age of 3, had antibiotics for 2 weeks to kill some of the bad strains first, and that would be of some help, especially as she saw an improvement in his gait going from this to normal, and also an improvement in les mots, in this language. So there was quite an improvement. La plus jeune fait quoi? L'epilepsy, it cured his epilepsy as well. So, merci. Thank you for sharing.
Okay, yeah, you're the last one, Emmanuel.
Just wondering, but why
I mean, the son of, the daughter, I don't know. So she had a diagnosis for the syndrome, but then why there is no communication between clinicians? That for me is something I really don't understand.
Yeah, it's difficult. And I don't, yeah, yes. Yeah, yeah, yeah.
I don't know.
It's, I'm sure, yeah.
Us as parents as well.
Yeah, so that's why there's a day like this to share, because also for me, there's a lot of, political issues and stuff involved in working together. And there is a lot to be done in that field of learning to work together better. I know that. So is it okay that we close now and go for lunch, Kelly?
Very good idea, because food indeed is important. We just learned. And so we will come back to this place. Okay, enjoy your meal.